Welcome to the Epigenetics Podcast from Active Motif. Join host Dr. Stefan Dillinger for lively discussions with leading epigenetics researchers. Hear about their past experiments, what they're working on now, and what's coming next. You know their papers, now get to know them and discover the stories behind the science.
Hello and welcome to this episode of the Epigenetics Podcast. Today I'm happy to welcome Vijay Ramani from Gladstone Institute on the show. Please let me briefly introduce you to our audience. You got your PhD from the University of Washington in 2017 and stayed there for a short postdoc. You then moved to UCSF, where you started your own lab in 2018, and since 2021 you are assistant professor at the Gladstone Institute. A question I like to ask every guest to start off our little podcast is, how did you become interested in biology in the first place and then in pursuing a career in science?
Yeah, great question. It's funny. My journey into biology. My parents were pretty keen on us having a lot of exposure to science, technology, medicine, to STEM, right? And I had some experience as a high school student, actually, when I was growing up, working at the interface of biology and engineering. I was just kind of working with a graduate student and a postdoctoral fellow at Rutgers University on some like very basic tissue engineering kind of research. I was a high school student. I didn't know very much, right? But it was interesting to kind of get my hands a little bit wet and get more exposure. But when I matriculated to university, my intended degree, and actually the degree I graduated, was chemical engineering. And so I think going into college, I really thought I was going to be an engineer. And then I stumbled upon a class that was being taught by David Botstein, who is a very famous geneticist and educator, who he created this. It was actually a multi-year course sequence at the university that was called Integrated Science, right? And the whole idea of it was that one shouldn't just learn chemistry, physics, biology in isolation, right? The right way to learn it, right, if there's a right way to learn something, right, is in an integrated framework, right, where you're taking courses in, say, computer science and then applying them to biological problems, right? You're taking classes in physics and then understanding how that applies to chemistry, right? And this kind of way of making these connections, right? Very interdisciplinary. And that course sequence got me very excited about quantitative biology. And so I ended up keeping my degree and my progress towards my degree in chemical engineering, but I started taking more biology classes through this integrated course sequence over the course of 3 years. And that got me really excited about biology. So that was really, that's what kicked it off. And then kind of kicked off with the career I have today. Yeah.
Yeah, I guess not to spoil too much of the content, but I guess there is a theme there, like an early theme where the engineering part still like resonates with you today, right?
I think so. I think it's necessary, right? I think so many of the problems that we and many others in the biological field at large are trying to tackle require one to think quantitatively, right? And so this kind of quantitative approach to biology, getting dropped into the deep end, as it were, as an undergraduate, ended up being quite useful later on. Yeah.
So maybe we can start with talking about your science. And I want to start at a time when you were in Jay Shendure's lab. Maybe we can start with the question how you ended up in Jay's lab.
Yeah. No. I recall. I wonder. I doubt Jay remembers this, but when I was interviewing for graduate school, I remember meeting Jay. Right? I remember meeting people in Jay's lab, right? And they were telling me about some of the unpublished things that they were doing, and this was during like the interview period, right, where you go and you visit the campus, that kind of stuff, right? And I was hooked, right? It's so interesting. It seems so in line with the type of science I wanted to do. I had been working for the past year at a company called Sangamo Biosciences. At the time, it was called Biosciences. Now it's called Sangamo Therapeutics. They were early pioneers of genome editing, and they were using engineered zinc fingers, engineered TAL effectors to effect therapeutic edits for human disease applications, and some of the work I was doing there, right, was at the interface of technology and molecular biology. And Jay, I remember him telling me during the interview that about some of the unpublished things and published work that they were working on, and I was hooked. I was so excited. I think I might have said, "I think I'm going to come here and I want to rotate with you," right? And I think he said, "No, you could wait. Hold on, you don't have to make any decisions, right?" But and then I did decide to go to the University of Washington and for my PhD, and I did rotate with Jay, and it was just it was, and I'm sure continues to be, a really spectacular place to do science. And it's one of the amazing places to do technology, right? And really do molecular biology in the service of technology. And something about that, when I was a first-year graduate student, really resonated with me. Jay also really gave us a lot of flexibility and freedom to go after the kind of methods and methods development problems that excited us the most, even if they weren't something that the lab was currently working on. And so I got a lot of, he gave me a lot of autonomy to work on kind of crazy ideas, and some of them worked out. So that worked out quite nicely. Yeah.
Yeah, let's talk about one of the things that worked out quite nicely, because you worked on a method called RNA proximity ligation, which, when I read the abstract, sounded pretty much like a 3C for RNA. Was this what you had in mind when developing this method, and how does the method work?
Exactly, right. So this, and I should say that, since we published that paper, I don't know how many years ago now, right? And that was the first piece of work for my PhD.
I wrote it down somewhere.
Howard Chang's group, several other groups have published similar approaches, right? That are even better, right? That use cross-linkers and stuff, right? But the idea was entirely, could we do high-C on RNA, right? In fact, I think I might have gone to Jay before my rotation and said, like, this doesn't exist yet. Could we do this? could we actually use proximity ligation to resolve RNA structures, right? And this began as like a rotation project that I just kind of started on a whim in Jay's lab. Around, by that point, high-C was pretty popular, right? I think when I was a first-year graduate student, the first high-C maps from bacteria from the Laub lab were just published in Science. TADS came out around then, 2012, I think, was Alfege and Jesse Dixon's papers came out back to back right around then, right? So the idea of taking high-C and applying it to something else, right, was kind of an untapped area. Obviously, proximity ligation had been used for detecting protein-protein interactions. I think there were some successes with protein-DNA interactions. And so we did some of these experiments, and we sequenced these junctions, and we realized that RNA secondary structure was kind of popping out from this, right? And that ended up being the paper, right? We were basically able to show that just by doing native ligation, digestion and religation of native RNA species, you encode two-dimensional information, right? And we saw that as like a complement to a lot of the 1D structural probing approaches that already existed at the time. Yeah.
Was this also useful for doing like RNA-DNA interactions, or just for RNA-RNA interactions?
Shang Zhong's lab has shown basically that it works now, right? So these like Margi protocols, and they have all sorts of names now, right? But in the ensuing years, many groups have basically shown that you can use nucleic acid ligation, right, of like cDNA to DNA, right, cDNA to cDNA, right, RNA to RNA, and then cDNA, which is what we did, right? And then there's also ligation-free approaches, right, that others have developed that are useful for this, right? It's a very hot area now, right? Like RNA-RNA, RNA-DNA interactions. I think still an open question what these RNA-DNA interactions might be doing, but definitely a lot of excitement there. Yeah.
If you can look at them, you can study them and maybe find out what they are doing.
Exactly.
So what you then did is you worked on improving the 3C protocol by including DNAs in the workflow. This was published in 2016 and is called in-situ DNase Hi-C. At about the same time, Olli Rando's lab published micro-C, which is Hi-C using MNAs. So a lot is going on at that time in the 3D genomic space. So did you also try MNAs, and how did in-situ DNase Hi-C work, or was it just like that you knew that they were working on MNAs and you wanted to do something different, or was it just like it works better using DNAs?
We didn't know that that Ollie's group was working on MNAs at the time, right? My recollection was that that the original version of the DNase protocol, right, which led to that Nature Protocols paper, it was a Nature Methods paper. I think that paper might have been in review or in press right around the same time as Ollie's cell paper. It's been a while. I don't remember exactly what the timeline was.
Yeah.
We had already begun applying it towards other problems, though, namely X chromosome inactivation. And so when I was doing those experiments, we basically had a protocol that we had optimized that was working really well. We weren't really interested in nucleosome resolution contacts, though, right?
Mm-hmm.
So I think that the problems that we're going after were slightly different. The only thing we were really interested in was trying to get a better Hi-C protocol, right, that would have less bias and would give you more cuts per bin, right? So I can't remember what resolution we looked at in the genome biology paper that was published, but it wasn't kilobase resolution, right? This was also right before the kind of kilobase resolution Hi-C maps started to come out, right? So this was an interesting time, right? There's like a lot of like methods development going around, right? Kind of different silos, right? But also people going after different problems, right? And so, so we weren't really. If you can even look at the kind of biology that people were going after, right? They're just different problems, yeah.
So it was more like tailored to the biological problem you were tackling, that you used DNAs compared to MNAs.
Yeah, and I should say that I worked very closely with a research assistant professor. He might be a research professor now at University of Washington, Jay Shendure, who has done like a lot of chromosome conformation capture methods development, dating back to collaborations he had with Stan Fields on mapping the structure of the yeast genome. So this was a collaboration that was kind of baked into the department at the time already. So I was working very closely with Zhizhen on a lot of this. And so the original DNase Hi-C protocol Zhizhen had developed. And actually, I think people don't realize this, but one of the first capture Hi-C protocols is in that Nature Methods paper and Zhizhen really optimized that, right? I think it's a little bit undercited, actually. Yeah.
Yeah, I mean, that's often a problem, right? So I guess it's clear now that this interview will be very technology heavy, because the next paper question introduces already a new method again. This time you introduce MNase SSP, a single-stranded sequencing library preparation method for nuclease digested chromatin that enables simultaneous mapping of transcription factors and nucleosome positions, which seems to be very helpful and also very interesting. So how does this work?
Yeah, so I think this opens. You point out something really interesting about my graduate work, which was that it was like a lot of technologies, right? Like proof of concepts, right? I think we use the word proof of concept a lot in the papers I used to publish, right? Right. But the goal was this, and it's still a goal that we still share kind of in terms of the work that my lab is doing now, right? All methods have bias, right? And so how can we rectify some of those biases and develop, a complementary method or an improved method, right, that allows us to get at more biology, right? And so so even with something like SSP, which is like a very small paper, right? One of the things that I had noticed, right, and others, this has been noticed in the literature for a while, actually, right, was that micrococcal nuclease has a fair bit of bias, right? But one of the really technical things, this is a very technical point, right? But one of the things that came up during discussions when I would talk to people in Jay's lab, when I talked to friends of mine in other labs at University of Washington, people in Steve Henikoff's lab down the street at the Fred Hutch, right? What amount of that sequence bias was coming from micrococcal nuclease and what amount of that sequence bias was actually coming from other sources, right? You could imagine that even like T4 DNA ligase, right, or the end repair reaction might introduce terminal biases that could alter the biology that you're trying to measure, right? And one of the interesting things from that, again, very small paper, was actually finding that you could decouple the micrococcal nuclease cleavage bias, right, from what are clearly other biases from other library preps. We did a bunch of in-house micrococcal nuclease, like traditional MNAC. We downloaded data from a dev cell paper from Ollie Rando's group, data from Jim Kananaga's group, right? Right, and we could see something really interesting, right? Which is that the shared library prep steps that all of those published data sets and the ones that we did in our lab, right? They were adding more bias, right, to the measurement, right? And just by changing the library prep, we called it, SSP is just short for single-stranded library preparation. It's a protocol that existed already that was used for sequencing very low inputs of ancient genomic DNA. And then my former lab mate, Matthew Snyder, adapted it for cell-free DNA. And then we just adapted it for micrococcal nuclease digestion products. It actually reduced the bias even further, right? And so those kinds of methods, right? I think DNase Hi-C, MNase SSP, right? Those were, I think, they ended up being a fair bit of work, but really just are trying to rectify biases, right? Like Hi-C has a restriction enzyme bias. How can we get around that, right? MNase library preps have their own sequence biases. How can we quantify that better and maybe even fix that, right? And I think that kind of mindset still, I think it's important for any methods developer to be thinking about, but it's something we still think about in the lab today.
How much does this bias then change the biological answer that you get? Is this I think, relevant? I mean, it's relevant for an academic.
It turns out it is relevant, right? Yeah. So I think here's the trick, right? Here's the problem, right? I think it's really important to be conservative in how we try to glean biological discoveries from biological data, right? And so when we think about bias, right, I think the way to think about it is that there are a set of, let's say, transcription factor footprints or nucleosome positions, right, that you might want to say are driven by sequence, right? But if you cannot distinguish that signal from the inherent bias of the prep, then you just can't make a conclusion, right? Right? And I think that's where the problem arises, right? Where you see this peak and you say, oh my gosh, that must be something biologically meaningful. But then, you have to do controls, right? And if your controls show that the extent of the bias is such that it matches what you're seeing, right? You're basically in a scenario where it's inconclusive. And so, even now with the methods we develop and we've been developing over the last 6 years in my lab, right? Like we think about that a lot, right? Like what is the bias of the enzymes that we use to mark chromatin and map where things are and where proteins are interacting with DNA, right? It's still something we think about a lot in my lab. Yeah.
Mm. I mean, we will talk about the methods you developed further along, but maybe we can take a detour now and because it makes sense now. So all the new, I mean, not all, but in the air quotes, right? The new methods that are very popular right now is cut and tag and cut and run. And all those methods rely on enzymes, not on shearing as it would have been with chip. Do you see this as a problem? Or should there be other controls be made on those methods?
There should always be controls, right? I think, look, I think it's really important to be very careful with how we describe the state of like methods application, right? There's a lot of excellent work being done with cut and run and cut and tag, even at the single cell level, right? And basically to a T, those experiments are incredibly well controlled, right? One does knockout experiments, right? One makes sure they use their, anti-GFP controls, obviously at a minimum, right? But I think that all assays have bias, and how one considers that bias is part of experimental design, right? So I think, are there going to be cases where one wants to do, say, cut and run, right, and is having difficulties where changing the library prep might actually improve the sensitivity for detecting a given peak or something? I'm sure, yes, the answer is yes, right? But what I have to think about, why is the bias occurring, right? What does the bias mean, right? And what is the right way to correct for that, right? Maybe it's a change in library prep. Maybe it's a genetic perturbation that can get you to your right answer. Maybe it's like incorporating a flag tag or something in your model, right? It really depends on the scenario, right? I think that especially when people come to say Active Motif and say hey, is my assay the right assay, right? Right. The answer is always it depends.
Exactly.
Right? Right. And I think that's a difficult thing to get across sometimes to the audience, but it's really important point to make. Yeah.
Yeah, there are many variables to consider when choosing the right assay. That's true. Next, to come back to the timeline, you moved into the single cell space and developed single cell Hi-C. So was this also one of those small papers you mentioned?
Yeah. So all of this happened in a 5-year span, right? Yeah. So this was between 2013 and 2018, right? I joined Jay's lab I was actually 2014. I started grad school in the fall of 2013, and I joined Jay's lab in January of 2014, I think. So it was all happened very fast. So there's a lot of stuff happening in parallel, right? I think SSP ended up being the latest paper that was published, right? So I worked on, to keep it quick, right? I worked on single cell combinatorial indexing, or SCI, this kind of new approach for doing single cell genomics. And I did this. I worked on this with a lot of people, right? Really, I think the SCI idea, it could really be attributed to Jay and Andrew Adey and folks at Illumina. Darren Kusanovich and I and Jun Chao wrote a bunch of papers together. Darren's a PI at Arizona now. Jun's a PI at Rockefeller. Andrew's a PI at OHSU. The idea was could you? So at the time, this was 2014, 2015, maybe. All of the published single cell genomics approaches all involved isolating single cells, right? So you'd use a sorter, right? Or you'd mouth pipette, right? Cells or nuclei into wells, right? And then do some kind of high gain amplification to amplify up RNA or DNA or what have you. The timeline is fuzzy right now, right? But some of the microfluidic approaches came out, I think in 2016, but I could be wrong. And so we were thinking at the time, wouldn't it be awesome if you could do single cell genomics without ever resorting to single cell isolation, right? And Illumina had a technology, right, that had been published, right, that allowed for basically the sequencing of single molecules of DNA without ever resorting to single molecule isolation. And they called the CPT-seq. And they would use basically a combinatorial indexing scheme with transposases to uniquely barcode individual haplotypes, right? And the idea was, could you basically extend that to single cells? This is an easier problem, right? And so we basically worked together with Illumina and figured out the molecular biology to do this kind of combinatorial indexing-based approach for a bunch of genomic assays. So ATAC-seq, that was SCI ATAC-seq. DNA-seq came from Andrew Adey's group. They call that SCI DNA. My PhD project, one of my chapters of my thesis was on single-cell Hi-C. So we were able to generate some of the first. It was I think at the time we published, there was 16 single-cell Hi-C maps, and we added several thousand single-cell Hi-C maps to that, right? And we could do this by basically figuring out how to incorporate barcodes into intact nuclei in small subpools. We then pool all those together, split them out, redistribute them, barcode them again, pull them back together, and then sequence, right? And the unique combination of barcodes, you can do this for 2 rounds, you can do this for 3 rounds, right? They basically addressed in each individual cell, right? And so now a company called Scale Biosciences is now marketing this for ATAC-seq and for methylation sequencing and for RNA sequencing as well. And so if listeners are interested in that, I think a lot of people are having success with those kits. Yeah.
So all we talked about now, and you alluded to this just before a little bit, was work that came out of your PhD. I must say this is very impressive. And you also said that you were working on all of this like in parallel, or at least in a short amount of time. How do you look back at these years? Can you tell us the secret to your productivity and being so innovative?
I worked with really great people, right? One person who is on every one of my papers was a senior scientist in Jay's lab, is Rulan Q, who recently retired a few years ago from Jay's lab. Well, she actually retired a while ago, and then loved science so much she actually came back to like work part-time in Jay's lab, and that's when I met her. And then I think she formally retired recently. She taught me how to do molecular biology, right, and how to do all of that, right. So and how to like, do experiments properly, right, and that was the big part of it, right. A lot of luck, but really working with amazing people like Jay at the top, but also just he really created a really fantastic environment where people were going after hard problems. People were unafraid to develop new technologies, and people were also unafraid to kind of ask the hard questions of technology development problems, right? I think like a really important question to ask when embarking on doing technology development, right, is like, why? like, is it like, does this problem really need to be solved, right? And that's like an important question to ask, right? Because there's like a bunch of, I think, technologies that one could develop that don't really move the needle. And so it's probably not worth going after that. And I was lucky to have like really fantastic lab mates who would, like, we would push each other, right? And ask like, do you really need to do that, right? And that kind of feedback really, I think, allowed us to focus on the really important problems. And then I think the kind of the proof is in the pudding. But yeah, it was a really spectacular environment to just work in. Pretty intense, but also really fun.
I mean, I guess what we talked about were 4 successful projects and 4 successful methods, but have you ever like worked on something that didn't make it into a paper? And when was then the point of no return where you said, well, I got to stop now or do it until the end?
Yeah. Yeah. Spent a long time, right? A lot of hours in the lab on a method that now exists, right? The idea, and it's nice to see it that's been published by other groups, honestly, right? Because it kind of shows that the idea was a good idea, right? We were trying to, in the early days of, a friend and I were in the early days of actually PacBio RS2 sequencing, right? Might have been the RS at the time. We were trying to figure out if we could come up with amplification-free methods to target specific regions of the genome, right? And so the idea was, could you use like in vitro Cas9 digestion to do this, right? And so we came up with protocols to basically do in vitro Cas9 digestion and like pulse field gel electrophoresis to try to isolate these fragments and then try to put them on the sequencer, right? Now that approach exists, it's called Catch, right? Mm-hmm. And many groups have really, Howard Chang's group has published some really nice work on using this to isolate ecDNAs and then put them on long-read sequencers. Sage has made a device that really facilitates this, makes it a lot easier. But it just never worked for us. We'd never get it to work. We would get digestions of like lambda DNA working perfectly in plugs. We could isolate, we could write it. It was cool to learn how to do some of these like classic techniques, but it just never worked. Mm. And we tried it. And I just think sometimes it's just how it goes, right? Like it's just sometimes molecular biology is like that. And so that's the one I remember clearly. There were a few other projects we just dropped, right? Yeah. But this one I remember spending hours in the lab, right? And eventually just being like, okay, like other stuff is working. We're just going to work on that. Yeah.
So you then started your own lab, and your drive to develop new methods did not stop. And you, and I guess your team, developed single molecule adenine methylated oligonucleosome sequencing assay, short SAMOSA. Nice name. Can you talk about the transition from Jay Shendure's lab to your own, and then about how SAMOSA works and what does it enable to do?
Yeah, so in 2018 I started my own group. It was through a really unique program at UCSF that allows graduate students to start their own lab right away. This independent fellows program, it's called the Sandler Fellows Program, and they give fellows 5 years of support. It's roughly an R01's worth of funding to basically run your own group, right? And when I started my own group, the single cell space was pretty crowded, and I felt really proud of what I'd been able to accomplish in that space, but I knew I wanted to kind of move in a different direction and also get a bit more mechanistic and really start asking really deep biological questions in the chromatin field, right? And really start to think about chromatin structure and the factors that drive that regulate chromatin structure in a mechanistic way. Around that time, PacBio sequencing was becoming really good. I had kept in touch with people at PacBio, even from my time in Jay's lab, and was hearing even just through the grapevine that sequencing quality was getting better. It was right around the time that circular consensus sequencing had become a thing, where the realization that rather than try to sequence really long, highly error-prone reads, one could actually leverage the fact that PacBio sequencer sequence the same single molecule over and over and over again to get really, really high quality data out of the PacBio sequencer. And the Sequel 2 had come online. And so, and I had prototyped some experiments in Jay's lab where we were using kind of non-destructive methods, using adenine methyltransferases to try to mark accessible DNA in the context of chromatin, and then throw that on a sequencer, and then leverage the fact that the PacBio sequencer could natively detect those methyladenines. That was actually one of the things that PacBio knew for a long time it could do really well, was detect adenine methylation specifically, to try to see if we could like non-destructively map chromatin accessibility. As it turns out, several other people were working on this around the same time, with both PacBio and with Oxford Nanopore. And I always like to say, like, there's nothing new under the sun. So there's always people working on similar ideas. But we had the good fortune of really Noor Abdulhai and Colin McNally, who were the first two trainees in my lab. They worked super hard, and they were able to get the method kind of up and running. And we were able to publish right around the same time as several much larger groups, right? So John Stam's group, Will Greenleaf's group, Winston Timp's group as well. And so the approach, all of the approaches are all conceptually very similar, right? Use a non-destructive approach, enzyme, so a methyltransferase, to mark where every nucleosome or transcription factor is on intact chromatin fibers, and then use a third-generation sequencer to read out on long reads where all of those installed methyl marks are, right? And this is also just built on footprinting-based approaches.
So would you, sorry to interrupt, but would you mark like the position where the transcription factor is, or would you mark everything but the position where the transcription factor is?
The latter, right?
Yeah.
So you're effectively marking all of the accessible DNA, right? So the unmethylated regions are the footprints, right?
Yeah. Okay.
And people have done this with GPC and CpG methyltransferases as well, right? And so, but the problem with that, right, is the resolution, right? So you can just compute, right, what is the density of CpGs and GPCs in a genome, right? And it's not infrequent, but it's not frequent either, right? Whereas A's and T's are quite frequent. And so we and others have leveraged that to get really high resolution maps of where proteins are interacting with DNA on intact molecules. Yeah. Mm-hmm. And that basically powers the lab now. Everything we do in the lab is revolves around this SAMOSA assay that we published now 4 years ago. Yep.
Yeah, I wanted to ask, because in the time since publishing the initial paper, you worked on refining it, or let's say to adapt it for other use cases, right? To name then it was SAMOSA chat or SAMOSA tag. So could you talk about the process thinking about SAMOSA enhancement and how those methods present an advantage over the classical SAMOSA?
Yeah. So we developed the approach, right? And one of the discoveries that came out of that paper that we published was that chromatin is really heterogeneous at the single molecule level, right? So you could look at euchromatin or heterochromatin, and you find that if you look at the nucleosome repeat lengths at the single molecule level for euchromatic fibers and heterochromatic fibers, they're different. They're significantly different, but their distributions overlap tremendously. And that we've confirmed that in every cell type we've looked at in the ensuing years since we published that initial finding, right? So then the question is, what's driving that heterogeneity, right? And like, what structures chromatin at the single molecule level, right? And this gets down to mechanism, right? So Noor, who was the first PhD student in the lab, she was the first author on the original SAMOSA paper, and then at the same time, she was working closely with our long-time now collaborators in Geeta Narlakar's group to see if she could apply SAMOSA in vitro. So what do I mean by that? Really, she was working with the in vitro biochemists and biophysicists in Geeta's lab to create chromatin in a test tube. She would basically dialyze nucleosomes onto physiological sequences. She would clone mouse sequences from the mouse genome and then put nucleosomes onto those sequences and then use those in vitro reconstituted chromatin fibers as a substrate for chromatin remodeling reactions. And then use SAMOSA as a readout to figure out what's happening at single molecule resolution. What are those remodelers doing to those chromatin fibers at single molecule resolution, right? And so we thought that we were spicing up the SAMOSA a bit. So we called this in vitro kind of cell-free genomics approach, right? SAMOSA CHOT, or SAMOSA to test chromatin, to measure chromatin accessibility on assembled templates. So sometimes I forget the acronyms too. So that was the SAMOSA CHOT assay, which we've been able to apply it in all sorts of settings. We had another preprint, again in collaboration with Geeta's group. This was a work that was led by Camille Moore, who was a joint student between my and Geeta's lab, where she could use the SAMOSA CHOT assay, combine that with imaging to ask what are the single molecule chromatin fiber structures associated with mesoscale chromatin condensation, and then how do remodelers affect that. And then we have some unpublished work that I've been talking about at meetings recently, where we're reconstituting even more complex chromatin biochemical reactions like replication, right? And then footprinting those. So the SAMOSA CHOT assay has been a big part of the lab over the last few years. SAMOSA tag, that came out of again, like a methodological necessity, right? So all of these single molecule sequencing based approaches, they inherently require the sequencing of native DNA, right? You're detecting the modification that you're installing on the molecule. So if you did any PCR to amplify up that molecule, you'd lose the modification, and that's problematic, right? So, but it also means that some of these assays aren't really extendable to clinically relevant settings where you might have very low input amounts of DNA. And so my longtime collaborator, Siva Kasanathan, and I came up with some ideas, and then Scott Nunda, who is another graduate student in the lab, really worked closely with a couple of techs in the lab, Ira Irklienko and Coco Wu, to figure out a molecular biological protocol where we could use hyperactive TN5 transposition, kind of like Nextera, but for PacBio sequencing. So we adapted, right, it really was an adaptation. We figured out a totally new molecular biological protocol. It actually uses a different TN5 transposase that has one more additional mutation to tagment DNA and then put that DNA on the PacBio sequencer. And so now we can sequence like nanograms of input DNA, PCR-free, on a PacBio sequencer. And so that, of course, extends SAMOSA to more clinically relevant samples, right? And then, so now we're thinking about how we can apply that to the study of like patient-derived xenograft models, for instance. Yeah.
And more recently, and this was the last paper I wanted to talk about, and this was published some weeks ago in November 2024, you presented replication-aware single molecule accessibility mapping called RASM. So again, is this based on SAMOSA, and how does it work?
So we take the same single molecule footprinting approach, but now we're adding another omic measurement to the single molecule detection paradigm, right? So we use the PacBio sequencer in SAMOSA to detect M60A and infer inaccessible footprints from that, right? Now we've also trained convolutional neural networks to detect BRDU incorporation on those same single molecules. So we can feed cells BRDU that'll get metabolized into BRDGP and incorporated into nascent DNA. And we can now distinguish in silico the footprints that come from BRDU positive molecules from molecules that didn't incorporate BRDU, right? And that effectively allows us to measure what the chromatin fiber looks like immediately behind the replication fork. We pulse for 15 minutes, right? We try to purify nuclei as quickly as possible, but of course, we might get a little bit of more fork progression as we're doing this prep, right? So we think we're maybe capturing chromatin fiber structure, we'll say like 50 kilobases behind the replication fork in embryonic stem cells, right? And that led to a bunch of discoveries that are detailed in the Cell paper that we published online a few weeks ago. Yeah.
I wanted to come back to something that you said 10 minutes ago, roughly, because you said that there is heterogeneity in hetero versus euchromatin, right? And they might overlap. And you said you wanted to go to the initial sources behind those differences, but what might be the biological consequences of those heterogeneities?
Yeah, so what's the consequence of having that heterogeneity? You think about what's the cause of the heterogeneity, right? We think it's ATP-dependent chromatin remodelers. It could be ATP-independent remodelers. In heterochromatin, it could be things like HP1. In euchromatin, it could be a number of things. It could be acetyltransferases. It could be HDACs, right? Same thing, same heterochromatin, right? What is the consequence of that heterogeneity? It's something we've been thinking a lot about, right? So I think what's coming out from our work, it's coming out from other groups as well, right? And I think even some of the electron microscopy, right, and that's being done on chromatin is suggesting this as well. Is that chromatin is a highly dynamic entity, right? So then the question is, how is regulation happening? How is repression, for instance, happening in a context where you actually have a lot of DNA accessibility, right? So we interpret some of this heterogeneity in heterochromatin, euchromatin to mean that euchromatin, some fraction of the time, has arrays that are closely spaced, right? Which I mean nucleosomal arrays where the nucleosomes are on average closely spaced together, right? Conversely, heterochromatin also has arrays, but where the nucleosomes are widely spaced or where they're irregularly spaced, right? So then the question is, how does repression happen in the context of that higher amount of DNA accessibility? And then how does activation happen in euchromatin in the context of that lower amount of accessibility, right? And I think that's something we're still trying to get to the bottom of, right? We know from a paper we published in NSMB, the SAMOSA chat paper, actually, we also did a lot of in vivo in cellular experimentation, right? We can show that the heterogeneity that we see in heterochromatin is at least in part consequent to remodeling by imitation switch remodelers. So the imitation switch family remodelers that we've known for a long time, co-localized within the heterochromatin, are creating some of that heterogeneity, right? They're probably dynamically moving nucleosomes back and forth, right? And creating irregular fibers that for an instantaneous measurement look irregular or look widely spaced, right? But what that means in terms of regulation, I think, is really interesting to think about, right? One model we'd like to test, right, is that what's actually happening is that what's really driving regulation is the nucleosome density, right? You have more nucleosomes per unit length DNA in heterochromatin, and so the probability that, say, like a Krebs motif is exposed is just statistically lower, and it's kept at this low threshold to prevent ectopic, activity of an enhancer, right, or ectopic binding of your favorite transcription factor, right? These are difficult models to test, but we're actively thinking about how best to test them. Yeah, does that make sense?
Yeah. Just thinking about molecules like pioneer transcription factors, right? How they come in. Probably those might be the first ones to act on those kind of heterochromatic irregularities and things like that.
Exactly, right? Maybe that's the state, that's the single molecule state that certain transcription factors or certain transcriptional regulators are able to take advantage of to drive a cell fate decision, right? These are precisely the models we'd love to test, right? And the devil's in the details, right? Because you want to do the experiment in the right way. Yeah.
Definitely. So imagine you're due to submitting a grant proposal tomorrow. What would you have written into this?
Yeah, not tomorrow, but soon, right? We're really excited about thinking about how we can dissect chromatin structural heterogeneity at the single fiber level, both through bottom-up approaches and top-down approaches. What do I mean by that, right? I say top-down approaches, I really mean like kind of candidate genetic genomic approaches, right? So we can take cells, we can engineer those cells with small molecule sensitive degrons, right? Where we can add a small molecule and very rapidly deplete that protein at the protein level, right? And then use assays like SAMOSA to ask, how does that factor directly contribute to the distribution of single molecule states that we measure, right? But while powerful, those kinds of approaches can be limited because of the inherent redundancy of chromatin regulatory networks, right? So I get rid of one remodeler, another remodeler comes in and does its job, right? You poison BAF, and then TIP60 comes in and starts moving nucleosomes, right? It's a real pain in the butt, right? To try to get around this redundancy, right? But this is where biochemistry, I think, is both elegant and really powerful, right? Because we can use our reductionist, reconstituted systems to try to complement the top-down genomic approaches, right? And so we can then use SAMOSA chop to say, okay, we have chromatin fibers for 100 for one sequence, 50 sequences, 100 sequences, right? We have nucleosomes that are unmodified, modified, right? We add purified transcription factors to the mix. We add remodelers to the mix, right? And then we ask, what do we see, right? And what is the minimal set of proteins, the minimal complement of regulatory factors and sequences required to get us close? We'll never exactly mimic what we see in vivo, I think, right? Or exactly mimic the distributions, right? We'll probably get pretty close to the averages, right? But we probably won't exactly mimic the distributions, right? But the closer we get, the more we understand about what the individual contributions are of each of those constituent factors. And so we think this kind of two-pronged approach to really dissecting chromatin-based regulatory mechanisms is going to be really powerful for the foreseeable future.
Yeah. So for the last almost 50 minutes, we've been on a journey through your scientific career. Did we miss something important, or would you like to add something?
I don't know. I think, so a question I sometimes get, right? It's like, how do you like balance, like what fraction of your lab is like methods development versus biology versus something else, right? Maybe like translational, right? And I would say like we do all three, right? But like the focus really shifts depending on the needs, right? I'd like to say, and I think what I'm about to say is true, right? We're very passionate about getting to the bottom of really understanding how chromatin works, right? And understanding how transacting regulatory factors interact with their cognate cis-regulatory elements to drive cell type specific transcriptional regulatory programs in health and disease, right? That's the overarching problem, right? I think within those there are a bunch of really interesting sub-problems that I think different people in the lab have their own different passions that they're going after, right? Whether it happens to be about chromosome organization or chromosome choreography, or how a given enhancer or set of enhancers drives oncogenesis, or how a set of transcription factors may or may not drive a very specific developmental program, right? But I think what really motivates a lot of the questions in the lab, right, is the kind of question of like, where are we resolution limited, right? So you could think of all of biology as this space that we're illuminating with lamps, right? And the lamps are the methods, right? And so, the area of biology that we can discover owing to the methods that we currently have is like kind of you could think of it as this one big circle, right? So then I think it's really fun to think about, okay, what's outside that circle, right? Like, what areas of biology are we just missing because we don't have the right methods to resolve that potentially exciting, new biological paradigm? And so I think that's where certainly, I like to think we're going, right? And how we think about this intersection, right, of methods and biology and translation, right? It's not like siloed, right? It's not, oh, this chunk of the lab works on methods and this chunk of the lab works on biology and this chunk of the lab works on how it translates. It's all kind of intermixed, right, as it were.
So my last question, and it's maybe split in two, would you consider what you are doing to be epigenetics? And if so, what is your personal definition of epigenetics?
I haven't read this new Trends in Genetics, so it's on my. I saw it. It was posted this morning. But so look, I think this problem of replication is epigenetic, right? I think the problem of how in dividing cells, states are faithfully inherited or lost, right, is true epigenetics, right? It's a very specific type of epigenetics, right? It's the propagation of epigenetic memory across mitotic divisions, right? But it is epigenetic, right? I would say that the vast majority of what we do is epigenomic, though, right? But I do think, especially as we've now and a chunk of the lab is now thinking about these problems about persistence of memory, and how chromatin fiber structures, whether they're memorized or not, right, across divisions. That's bona fide epigenetics, right? It's not transgenerational epigenetic inheritance, right? As some people, as the epigenetics true believers might, really harp on, right? But I think that's the one aspect where we are thinking about like epigenesis in that way. But no, most of what we do is epigenomic technology development, right? Okay. It's molecular methods development with a focus on epigenomics.
Okay. Yeah. Okay. Thank you. And thank you, Vijay, for your time and for being on the show.
Oh, yeah. Thank you. This is fun.
Thanks for listening to this episode of the Epigenetics Podcast from Active Motif. We hope you enjoyed it. You can find all the mentioned references in the show notes. Please rate, review, and subscribe to our podcast on your favorite podcast platform so you never miss an episode. We'd love to hear from you, so please send us your feedback on Twitter, Facebook, LinkedIn, or via email at podcast@activemotif.com, and we'll give you a shout out in a future episode. For more great epigenetics content, check out the Active Motif blog at activemotif.com/blog. Thanks for listening and stay tuned.